So good afternoon, everyone, here in the room and also online, and welcome to the presentation session 2, Data and Research and Data Science and Data Analysis. My name is Maja Dolinar, and I'm the director of the Slovenian Social Science Data Archive from the University of Ljubljana, and I'm really delighted to chair this session today. So before we begin, I would just like to acknowledge the traditional owners, the Turrbal and Yugara peoples of the land that we're meeting on today in Brisbane, and pay my respect to the elders past and present. So today's session will bring together, as you will see, a diverse group of researchers and practitioners addressing one of the central challenges of our time, so basically how to make data more meaningful, interoperable and actionable across scientific and institutional contexts. We will explore, as you will see, innovations in genomic infrastructure, healthcare data integration, data stewardship, AI and machine learning for text and anomaly detection, and the governance of federated research platforms. Please remember that this session is being recorded and live streamed. We ask that all participants follow the conference code of conduct, and that you submit questions via the event app, either for virtual attendees and also people here in the room. And of course, you will be also able to post your questions later on the Q&A session in live. Each speaker will have 10 minutes to present their paper, and I would really encourage all the presenters to be really respectful of this timing, because we have 8 presentations and only 1 hour and a half of time. So that leaves us, if we're really good, just 10 minutes for all the questions and answers. So today we will be starting with the first presentation by Dr. Kathryn Hall from Atlas of Living Australia. So let me just open this presentation. Okay, sorry. Where is the mice? Okay. Oh my God. Okay. So her presentation is titled Australian Reference Genome Atlas, Supercharged Exploratory Infrastructure for National Scale Genomic Data Discovery.
Thank you, Maya. That time gets shorter every time you turn around. I thought it was 11 minutes before, now it's 10. I've already timed this talk and it's way too long, so let's make it a whistle-stop tour. I'm sure Maya will card me when I'm going over length. So, yeah, it's really lovely to be here. Unfortunately, I've got the after-lunch session, so I've got to be the song and dance act to wake you up. So let's get going. The ARGA project is a project that's been done in partnership between the Atlas of Living Australia, the Australian Biocommons and Bioplatforms Australia. We also partnered with the ARDC, and as such we form part of the NCRIS infrastructure network within Australia to provide research infrastructure for Australia's researchers. So what on earth is ARGA? Well, there are about 15,000 life sciences researchers in Australia who rely on genomes and genetic data to get their work done. That's important work around improving livestock and crops for Australia's industries, but also about informing policy responses for our threatened species as they face increasing threats against things like climate change and, of course, bushfires. And if you were here on Saturday, you'd know just how hot it can get in Brisbane, very. But the reason this is a data problem is because the data that people are relying on are scattered, they're very diverse, they are across a number of international repositories, and they're quite hard for researchers to find, particularly as access to genomic resourcing or creating genomic resources is getting much easier. So there's a lot of it proliferating, which means that it's harder and harder for researchers to find data. The more time they spend finding data means less time for research, and also runs the risk of them actually wasting their precious funding resources repeating the efforts of other researchers. So the core problem that ARGA was trying to solve was a data findability thing, to create an access point for researchers to get the genomic data for species relevant to Australia's needs. So to do that, we wanted to combine the whole genome data and DNA sequences and other variant types of data with the animal and plant taxonomy, as well as specimens information and traits, to create a single interface where people could come and look through a number of different lenses to try and find the information that they wanted to solve specific real-world questions. To do that, we had to incorporate the genetic data, which is stored in places like GenBank using INSDC standards, but not all of the repositories we were looking at use those standards, or they may apply the standards variably. And we wanted to blend that with biodiversity data, which mercifully is mostly stored in Darwin Core these days. But when we were looking at it, we realised that there was another problem. So what is the data problem? Well, the problem is that Darwin Core makes the key data source that we were looking at, biodiversity data, quite flat. And I've got these two lovely ancient Greeks here because I wanted to give you a little bit of analogy. So in ancient times, we didn't know that the world was round, but the Greeks thought that it probably might be, and then Aristotle showed that it was. And that sort of opened up, I suppose, collective thinking to the idea that we might be able to traverse the globe. So in a similar sort of way, if we leave our biodiversity data very, very flat, it limits the possibilities and things that we can do with it. So what do I mean by that? Well, what I mean is that Darwin Core is actually really, really rich, and it's the main model that the biodiversity data are stored in. But what it does do is it relies on one material entity or a thing, and then it provides a whole heap of metadata descriptors. And as I said, they're quite rich, but it is this one-to-one relationship that causes the data to be quite flattened and not actually model the way that the real data exist. So what do I mean by that? Well, here's another complicated slide, not as complicated as the next one, but you don't need to worry about the detail. But the thing is that animals and plants have their own hierarchy that they use, which is the Linnaean hierarchy or classification system that they're stored in. And this is sort of derived through a discovery process. And we can liken it a little bit to semantic versioning, which is handy because there are things like synonymies and that sort of thing that account for name changes. So one plant may have a name this year, but scientists may use evidence to say that it's got a different name next year. And tracking that becomes quite a big problem and a big issue for people who are working in biology. And so that's something that we had to deal with. The second thing we had to deal with is, and this is a very detailed slide, and it's only detailed to show you that genomics data can be extremely diverse, numerous in type, and that they also have hierarchical relationships of their own, but also that they interact in the species space. So you could have genomic data derived from one specimen, but that may not actually be relevant for the whole species if the species were to be split, if it didn't come from the type. So there's a whole heap of complexity in there that we had to handle. And then the other thing, the final piece of the puzzle that made life complicated for us, was that genomes don't actually come from organisms. They come from amplicons generated in labs, and those amplicons come from tissue or from DNA extracts, from tissues, from subsamples, from specimens stored in museums or herbaria, which are derived from an organism in the wild. And what can happen along this chain is you can get duplication or replication, so that we've got these one-to-many events down the chain, so that any animal genome may have actually been derived from a couple of hundred amplicons, which is exactly what we see happen. So in reality, again, just another really complicated slide, because I just want to prove to you how hard this really was. But in this example, I just want you to see that the pathway to getting to the reference genome was very, very complicated. We had to go through quite a number of steps, and the little purple boxes are showing you just how many little data products there could be along the way that we have to track and integrate and make available to our users. So the challenge for us was to re-architect this wonderful, very rich Darwin Core model, but make it something hierarchical. The wonderful people at Tadwig who look after the Darwin Core standard do have an event model, but it wasn't quite nuanced enough for our needs. And we wanted to make all of the little puzzle pieces that are there findable for the researchers when they come to use ARGA. And so what we ended up doing was we created this model. Again, another very detailed slide. I don't want you to worry about it too much, except for suffice it to say, we broke it down into a number of steps. So our solution was to create a time series where we would go from the organism in the wild state through the various steps with accessioning and subsampling it to derive the tissues and the DNA, and then the amplicons and etc., and all the data objects that were downstream derivations from that to arrive at the final genome assembly. We also modelled in data reuse, just because we like to track reuse of any pieces of data that were found. So how did we do that? Well, we took an RDF ETL layer and we put that with a relational core, and we made it accessible through a GraphQL access layer. We maintained all the persistent identifiers we could get our hands on, whether they were DOEs for publications, whether they were specimen identifiers that were stable, ORCIDs for people who were doing DNA extractions, things like that. We created a DAG, or a directed acyclic graph, for our taxonomies, so we could align different schemas from different institutions or different organisations. Like the Atlas of Living Australia, where I work, maintains one taxonomy, but other researchers might use something like worms, and we wanted to be able to allow people to crosswalk those with ease. With ease, pardon me. How am I going? Oh, good, okay, I'm racing. So we made a custom Next.js interface, and that allowed us to basically expose a whole heap of richness in the genomic data and create context. I'm not going to have time to give you the full whistle-stop tour, so I've got this slide here to let you know that what we did include were genomics data, taxonomy data, traits data, and specimen-based data, so that researchers could answer very real questions in conservation, biosecurity, biodiscovery, industry purposes, and also biodiversity. And the main mechanism that we use for that is these traits views. So here's an overview of the threatened species index list that the Australian government has prepared for the top 110 species in Australia that are under threat. And we created these bespoke graphics where you can see the rate of accumulation of genomic data for them. You can see at a glance who's got genomic data, who doesn't have genomic data. We also created the same interface on a taxonomic level, but with a few tweaks for things like birds or any higher taxa. So you can also see there's a pinwheel on here that lets you know which groups within birds have got a lot of coverage and which ones don't. And there's a ticker there that tells you the rate of coverage of higher taxa. All of these graphs can be found for all of Australia's biodiversity on the genome tracker, and that has an accumulation plot for the rate at which Australian species are being having their genome sequenced. What was really cool about this and made it really possible for us was a very complex SQL query that we were able to build over our data model, but also the fact that in Australia we have the National Species List, and the Atlas of Living Australia hosts one copy of all of the species that are known. So we had a baseline that we could work from. Users are able to download all of our data in CSV formats in Darwin Core. And they can also access this sort of information for very specific groups and look for gaps in the data. So this is a picture of the mammals. Some groups have excellent coverage, some don't have a lot of coverage. Things like rats and bats don't have much coverage, which is quite interesting when you think about it, given how important they are for human health. So using a tool like this, we can immediately see where the gaps are, why those gaps might be critically important for us as a nation, and direct funding, and it fits into that. I'm not going to have time for them all, so you can come and look at my beautiful interface later on. But the only thing I want to say in summary is that the work that we've done is about creating context for all of our data. We had to amalgamate two very different sets and bring them into a rich narrative view where users could come and ask and answer real-world questions. And I think that's actually the best use of any index, is not just looking pretty and being able to find things, but actually using that information to answer real-world questions. Thank you. That was quick.
Thank you so much. Okay, so the next presenter is Ms. Nadine Andrew from the Monash University, and her talk is Creating an Integrated Electronic Health Record Data Platform for Revolutionizing Healthcare Research. Please.
Okay, thank you. I've also got a lot of slides, so I'll be doing a whirlwind tour, so please bear with me. So I would like to acknowledge the traditional owners, and particularly the Bunurong and Boonwurrung people, who are the custodians of the land on which the National Centre for Healthy Ageing, where I work, is. Okay, so digitalisation of medical records has created a lot of opportunities for researchers. There's some amazing data in there. It can be very granular clinical data. It's regularly updated, sometimes in real time, and it has the potential to enable us to discover and look at patients over their full journey across the healthcare system. However, there are a number of challenges as a researcher in using these data. In particular, they're collected for operational and clinical delivery purposes, and so there's little interoperability between the many systems that these data are collected in within a health service. Not a lot of standardisation, and a lot of the gold is in the text data in the clinical notes that the doctors write, and that can be very difficult to access. So we decided to try and solve this problem through an academic health service partnership between Monash University and Peninsula Health under the National Centre for Healthy Ageing banner. So why Peninsula Health? So Peninsula Health is in Victoria, down the bottom of Australia, and it's a peninsula. So that means that most of the people who live on the peninsula access their healthcare through Peninsula Health, which is the only public healthcare provider in the region. Peninsula Health also have a common UR number across all components of its health service. So it's acute hospitals, rehab hospitals, and community-based services. It has a reasonably large population, a lot of socioeconomic diversity, and it covers both some regional and metropolitan areas. We're interested in ageing research, and this is a really popular place for people to retire. So we have an older population. I'm just going to give you a brief overview of how we approached this problem that we were trying to solve, and then I'll go into a little bit more detail. So essentially, we wanted to bring together the data from all of the components of the health service. We wanted to organise it in a way that could be released for research in a streamlined and efficient manner. And so we developed a core data set, but we have all of the other data sitting around that can also be accessed on a project-by-project basis. We implemented CogStack, which is a natural language processing pipeline to give us access to the clinical text data. And then the other advantage of having a core data set was that we've been able to then link that to government data, to environmental data, and to bespoke systems, be them research projects or other healthcare provider services. So we started with trying to figure out what we wanted to put in our core data set. We started with a scope of the literature to find out how researchers were using electronic health data. We then got our base set, and we went through a data quality audit. So we tried to find those within our system and run them through this audit, which was a structured framework developed by Khan et al. We then chose to keep the data items that we knew had relatively good completeness and that either had good quality or we knew we would be able to improve the quality of those items. We then ran our big list of items through a Delphi process. So we engaged with clinician researchers at the health service and academics to understand which of those items they considered to be valuable, not just for their own research, but for research more generally. And we went through a consensus building process to come up with our final list of 131 data items. So we then went into building our warehouse. What we have ended up doing is copying all of the systems within the health service. And this is because we didn't want to impact on the operational capabilities of the health service, because we knew that would make us really unpopular and probably kill our project. And so what we're doing is taking copies of their data on a weekly basis, and then developing infrastructure to be able to then copy key clinical components of that into our staging area. This is our data schema. So we have basically 12 core modules of data sourced primarily from 4 systems, but then we've got another half a dozen systems that hold specialised things such as imaging or pathology or radiology. We've then created systems between that so that where we have an item that may be not well recorded in one system, we can supplement the quality of that item by drawing in data from another system. As I mentioned, we implemented natural language processing pipelines within our system, and this has been really important for us because I think this is one of the few, if not the only example of this existing within a health service. And what that has enabled us to do is generate new variables that may not exist in a structured format but are important for understanding health. It's helped us enhance the quality of our data where we know something may not be well recorded. And we're now starting to use it to develop sophisticated predictive analytic tools. And we've been able to show that when you use a combination of the unstructured and structured data, you're able to get a much better performing predictive model than if you use either of those two on their own. We've implemented software to allow us to do data linkage. So that means if someone has a project in our region where their participants haven't been recruited through the health service, so they don't have a health service medical record number, we can link them based on identifiers. And so that's expanded our capability, and we've done that for a couple of trials and cohort studies. So this is where we've landed. We've now got a curated electronic health data platform with over 1 million people in it over a 10-year period, so we're able to follow people over time, our 131 core data items, and we're updating that weekly. The next challenge is always data access and governance. So we've put quite a lot of thought into this. We wanted to be able to release data quickly because as someone who uses health data from government, it can take us sometimes 2 years to actually get the data we need for our research. So we're aiming to do a turnaround within a couple of months with our data. And we work closely with the Office for Research and the legal department at Peninsula Health to make sure we could release it in an efficient but a robust manner. And we've put a lot of thought into making sure that we comply with standards such as the 5 Safes framework and the other standards that are being used by our government organisations. Metadata is the other thing that we're working on. So we've developed a data dictionary for all of our 131 core items and spent quite a bit of time in that additional information section so that researchers can understand the limitations of the data and also, where the data have come from, how it's recorded and how that might impact on how they interpret their data. So we have so far supported over 60 different projects looking at a wide range of things. So we looked at health presentations during COVID, evaluation of community health programs, some environmental factors that might influence health, number of projects in opioid surveillance and substance abuse, as well as our algorithms that we've started to get some traction in. The other thing that's been great is we now have this really valuable piece of infrastructure which we've been able to leverage for other types of research grants. So looking at how we can use that to deliver better digital health, better monitoring, and we've got a smart wards sensor project happening as well. This is our future direction. So we're now partnering with the ARDC in the Social Science Research Infrastructure Network to look at how we can expand our data into the social science network to better examine that interaction between health and social science data and looking at how we can link our data with social science data held by government. We're also wanting to look at the feasibility of whether or not our model can be expanded to other health services or other projects across Australia. So if anyone's interested in being part of that, please contact me or our project manager because we're just about to start setting up some working groups around that as part of this ARDC project. So finally, thank you to our amazing team. And now you can see why everyone wants to retire at the Mornington Peninsula. So thank you.
Well, thank you very much, Nadine. And the next speaker is Hidetomo Kaneyama from RICAN RCCS, and his paper is called Study on Handling Dark Data in HPCI Shared Storage System Using the Wheel Workflow Tool. Here you go.
So I'm Kaneyama from RICAN RCCS. So I'm very happy to share our work, work here at the International Data Week. So my main task is operated supercomputer. So name is Fugaku, and storage system in Japan. So I'm a storage engineer. So today, I will introduce method of handling dark data in the HPCI shared storage systems. So first is overview. So outline. So we are operating the shared storage system service in Japan. So this service is provided free of charge to the research, so who use supercomputer in Japan. So as a result, so large capacity of data has been stored. So including many cold data. And so those cold data is also a lot of dark data. So dark data is unclear of information and unused data. So user does not understand the information, so such as so what is, and so why, and so who created, so what is value. So this is dark data. So because of this dark data, users don't remove unnecessary data. So our storage can't be used so efficiently. To solve this issue, so we are now developing a method to automatically annotation expanded metadata, so within our workflow tool. So next is this is the overview to the HPCI shared storage service. So this service is a national project in Japan, and so this storage was stored research data. So currently, HPC shared storage is running out of capacity, and user allocation are overcommitted. So and importantly, the HPCI shared storage is provided free of charge to research project. So this is free storage. So as a result, usually a way to clean up their data until the storage is almost full. So unnecessary research data has been stored for a long time. So we have this issue. So next is, this is graph shows the amount of the cold data of HPC shared storage. So here cold data is defined as data not accessed for 1 year. So as you can see, about 90% of data is cold. So this is very big programs. And some of cold data are dark data. So this is very bad things, I think. So we want to effective use of this storage. So we sometimes ask users to clean up and managing their cold data, but the user cannot remove data. So because HPC shared storage, the data and a lot of supercomputer storage, data information is unclear and unsettling. Don't data management, metadata management. So sometimes users can't understand themselves data's information. So such as what the data is food created. So users cannot judge. So if users can be safely removed and reused. So we think it's necessary to annotate expanded metadata to research data in order to reduce cold data and dark data. And we will promote the reuse of stored data for open science and AI, AI application or AI environment. So as one approach to cold data and dark data, we currently provide the user with visualization of storage usage. However, we recognize that the information we provide is not sufficient for users to manage their data in detail. Because it does not clearly show what kind of data and how much each of the type. Dark data continue to include probably our storage system, our storage service. In addition, we will connecting to the open science and AI environment near future. It will be necessary to handle metadata. I think we need to necessary, such as tags and labels and metadata management. We need. Now, in practice, many users in HPC of Japan are still manually management now. Such detailed metadata in like the Excel or text file. Very old type management. So we must change to the modern data management. So we think. So next is this is our approach. So okay. So therefore our approach focus on improving the user-side environment. So we extended the wheel workflow tool, so which is already used to on the HPC and the supercomputing system in Japan. So that if we developed new method on the automatically collected and auto annotation expanded metadata, so such as HPC name or supercomputer name and project name and software name and job ID. So and create time. So we added to the wheel the I/O API and also HPC shared storage in 2024. So we implemented automatic metadata collected method in this year. So next is a discussion slide. So our approach focus on the user side. But of course we now improving. So only the user side is not enough, I think. So ideally, data management should be consistent from the moment data is created, generation. However, changing the storage environment of HPC and the supercomputer is very difficult. So and also integrating new metadata system across all supercomputing sites. So would require huge cost and huge effort. So it is not realistic. So we choose user-side workflow-based approach. And next is user-side approach merit. So this allows us to manage data, not only the supercomputer. For example, pre-processing computing on the user environment, so we can use. Second merit, we can annotation, expanded metadata at the dataset level. So because we have the HPC shared storage have archive transfer method. The third merit is we have search CLI. So if data is expanded metadata stored in the HPC shared storage, so can be searched using metadata via the CLI command line. So to use the XML attribute special command. So sorry. Okay. So next is overview of the Wheel shortly. Wheel workflow tool. So Wheel is open source, web-based HPC workflow tool. So user can build and execute it their own HPC workflow on the web browser. So okay. So drag and drop and operated like this video. So Wheel is used Japan supercomputer and since it is open source. And so it is easy to extend it with few functions, such as the integration with the HPC shared storage and metadata annotation. So you can check the detail from the QR code. I'm sorry. I mistake. Oh, I'm sorry. I need the last slide. Can I back? Oh, I'm sorry. Okay, dash. Okay, this is final slide. So I'm sorry. So method. So this is first is method and future challenge. So a little time. So first is we created workflow with the Wheel workflow tool and supercomputer run and the pre-posting tasks. So when the workflow is executed, extended metadata. So and the software name and the job name and the label and username. So automatically collected during the computation. And the final step, so the output data is archived as a single set and uses tar or zip and stored at the HPC shared storage. At that same time, the extended metadata is also registered in the metadata database of the HPCI storage. It is very simple and very easy metadata management. So I'm sorry, sir, I have no time. So last hour, so future challenge is skip. I'm sorry. So this is future challenge. So okay. So the last is the keynote and the reference slide. Thank you very much.
Thank you very much. So our next presenter is Federico Grasso Toro, who's going to be presenting us introducing FJORD, a framework for fairly jointed open research data.
Hello, everyone. We're a little bit behind, and people that know me know that I speak a lot. So I will not go into details because I have a poster for this, so we can just sit down and have a long talk in the afternoon about this. So let's go step by step. I would describe a little bit of the problem that we are having from the point of view of current research information systems, and I will try to detail the framework as a proposal for the solution. Afterwards, I will just detail a little bit of how we have started with the idea, and I would just mention where we're going, because this is just basically the beginning of a proof of concept. So, as I said, the first current stage of things, as I call it, is that most systems worldwide, especially for research in general, are like little data silos that are not mainly are not designed to communicate to each other because they have been designed in different moments, developed in different moments, and with different status. So this is one of the big issues. The second issue, as a data steward, I have to say that most researchers use the data management plan as a very static document that is just basically something that they need to send to senders and not actually use it as a tool. So for those two things, the solution, for those two things, we just created a permanent issue here. That is, we have bigger administrative burden due to all of this, but also we created some sort of what I call poor data traceability. That is that when we only receive, when we only see the outputs or the intellectual assets that come out of projects, we have no idea how they've been developed, and we have no idea how the actual institution planned to build them. So this is a big issue, but as the third issue as well is that we are in the digitalization age. We're trying to digitalize research, but we're losing the greatest opportunity of actually creating machine-to-machine pipelines about specific things such as research reporting. So with that said, I make the joke there of saying, like, instead of searching for a solution, let's research for a solution. And what we have is we have two options, whether we add a new system. So we already have a lot of little islands. So we're suggesting we create a new island, and then that island would help all the other islands. Or we destroy everything and we start from zero from a current research information system. I doubt that any institution will be interested in the second one, but the first one creates new issues. So at the end of the day, we have a lot of little islands, everything is separated, and that's why I'm suggesting let's build a fjord. A fjord will connect all these islands by means of metadata, and it will not be an additional an additional system in itself. It would only be an orchestration layer by means of metadata. So just to describe it quite fast, I already said it, metadata layer that would be seated in between systems instead of actually creating a new system that brings new protocols and new issues. The idea then is that because we are thinking already in metadata level, we will have research activities that we can build into some knowledge graphs that are easily navigatable, because at the end of the day, most research tend to be structured or well-known from one generation to next in projects, so the idea is to learn from each. And at the end of the day, we have three main type of users, what we call, that would be like the researcher, the data steward that should be focusing on the open science of things, and then the funder that is just, they don't really fully understand what the project is, they just know grant, resources, and reporting. So if we put all that together, and I will move much faster on this, is from outside of the institution, we will have still the data management plans that we will make machine actionable data management plans, but inside the institution, we will have something that I already presented in previous conferences called enhanced DMPs that would include additional information that would be necessary for the PIs or the researchers to actually track the evolution of the assets or any kind of research output. So thinking of this, we need to be able to orchestrate persisting identifiers, and this I already believe is a success because in the morning we discussed about this with the research activity IDs, and there's going to be a new interest group in RDA about it, and I will be being part of it, because the idea at the end of the day is what we need is not just to report on persistent identifiers, but also to be able to advance in dynamic meta-persistent identifiers, such as the research activity ID. So with that, let's show you the win-win-win scenario that I'm proposing. At the end of the day, we will have a researcher that would have a simplified tool to actually manage the outputs of the project, with a logged, so being able to log the changes in their data management plans just as a platform, so they don't know that the whole thing is actually creating versionings of the data management plan, and a fully automated project reporting, because at the end of the day, once they've recorded everything, they just report on it. They don't have to write again a report for the funder. On the other hand, the same track of FJORD, but from the point of view of the data stewards, and this is what we are doing in the University of Bern, is we will have a way of tracking the actual fairness, so how findable, accessible, interoperable, and reusable these outputs are, and we will have the option of actually being flagged the different outputs during the project and not at the end of the project. So that would also save us a lot of time. And last but not least, we need to talk about the bosses. So both for management in the institutions and for the funders, they will have a very easy dashboard way of understanding how the already validated and assessed information of the output has been done by the people that actually understand about it. May that be the researchers, the PI, or the data stewards. So all of that would ideally set a machine-to-machine, machine-actionable data management plan between the institution and the funder. For that part, look at the poster in the afternoon, because that's the specific topic that I discussed about the common standards for the RDA. But now, let's just focus on what is on the other side, so on the side of the institution of what we are setting up. The first step that we've set up is, as I said, most people don't understand that data management plans are a very useful tools. So for that, we've created like a demo called Proposal Project Platform, that very quickly what it does is it asks 16 questions, and from those 16 questions, in the, let's say in the back end, creates a data management plan that is actually going to be evolved. So very quickly it is, maybe it's too small, but it's about the specific type of data that you will collect in, it's about the specific responsibilities and resources, and the type of documentation that we'll do. All of this, the researchers or the PIs feels even before writing the grant. Why? Because with this, we have the possibility of triggering infrastructure requirements, like, for example, high-performance computer research, before they actually need it. Stuff like open science support, like, for example, when there's sensitive data involved, the data steward will know before the report, before the grant has been applied, that they need to have support. And last but not least, if you need, for example, research software engineers, or any kind of additional support that the university should give you, we will know about this before you apply for the grant. So we will have a better way of actually planning strategy-wise. Okay, 2 minutes. So by that, right now what it works is we have these questions, and once the questions have been done, you can just basically download it as a TXT, because this is already your data management plan. Or you can submit it, and you submit it to our FJORD, and basically we break down the automatic request for example, for research storage, for HPCI, or so on. Because I only have 2 minutes, I will just jump saying, and all of this is integrated with the RDA, Machine Actionable DMPs, common standards. If you have questions, we'll talk about that later. And last but not least, the idea is we're starting with very basic setups and very basic pipelines, but the idea is to be able to scale this orchestration by means of what actually these three stakeholders need. And for that, where are we? Here. I have, if you want, this is a form, it's 5 questions. You can answer the questions as a type of stakeholder that you are. May that be a funder, may that be a researcher, may that be a data steward. And you can mark that you will be interested in knowing how this evolves, or you can mark that you'll be interested in doing a workshop next year, because we will orchestrate an online workshop with different stakeholders from different parts of the world to see if we actually advance on the thing. Thank you so much.
So thank you very much. The next speaker is Thabo Kumaran from the Architecture Victoria Research Department of Energy, Environment and Climate Action of the Victorian State Government, and the paper is Beehive Health Patterns Using Multimodal Data Analysis and Unsupervised Machine Learning.
Good afternoon. So I'm Thayalakumaran. I'm presenting on behalf of the main author on the Beehive health patterns using multimodal data analysis and unsupervised machine learning.
Can you speak in the mic, please?
Sorry. So the 1/3 of the Australian food sources are dependent on the pollinators, such as the bees. Particularly the horticultural industry, such as the almond industries, the summer fruits, and the apples and pears. So they're all dependent on the bees for pollination and the production of the fruit setting and the production of the fruits. So therefore, the beehive health is critical for maintaining the health ecosystems and supporting agricultural pollination. So if there is a decline in the bee colony vitality, that can negatively impact the crop yield, and also it poses biosecurity threats. Some of the main problems that we see in the beehives are the pesticide usage, then the varroa mites, then there are pests and diseases happening within the hive itself, and also the extreme environmental conditions. So there are some companies provide sensors to get the data within the hives itself, but they come with the proprietary models or algorithms which are difficult to understand, and also the scientific validation is not very open. So therefore, this work was conducted to enhance the accessibility of the data, analysis of the data, and then also to provide visualization of the whole set of data that we collect, and also to provide a method that's scientifically validated as the beehive health is understood, and also the method is kind of providing a warning signal for the farmers and also the apiary industry as such. So the data was collected in a target area, because this is more conducted as a proof of concept or pilot study. So it was more focused in the almond farms in Sunraysia region of Victoria. And then we collected multimodal data from about 3 farms at this stage. And from each farm, there were 6 beehives that we collected data from. The time period was just about a month during the pollination time in August to September. And as you can see, there were like multimodal data that were collected using sensor data such as the temperature, pressure, humidity that affects the hive health. And these were in multiple times in a day, like in 10 minutes intervals, and they were provided in kind of CSV files. And then the acoustic data, that's the sound energy within the hives, that was also collected in the waveforms, and again, it's in multiple times in a day. Then the agrochemical data that were applied within the farm, and they were collected in the frequency, how they were applied, and they were provided as in Excel format. And then there is a human inspection data. These inspections were done in the hives by farmers themselves, or some scientists went there to inspect what's happening in the hives, such as what's the queen presence, the bees' energy levels, and if there are pests and diseases that were observable there. and those are the visual inspections that were done. And also there was debris collected from the hives, and these samples were sent for eDNA analysis, and from the eDNA analysis we were able to interact with the, integrate with the other data system to get the anomaly system developed. So then to make sense of all this data, we applied some standardization of the data, and then data analysis and visualization of the multi-sensor data that we received from the hives. And also for some of the acoustic data, we have to apply some feature engineering so that the raw data can be changed into a feature that can be understandable and also can be used in the multimodal analysis. So in terms of the machine learning approach that we applied, we have to apply an unsupervised machine learning approach because the data, the hive health data wasn't labeled enough. So we have to apply the unsupervised machine learning rather than a supervised machine learning approach. So here we used clustering to see whether there are any clustering in the hive groups. And also we applied the anomaly detection using about 3 algorithms to see whether there is consistency in what we see as anomalies. And then we evaluated the anomaly detection using the human inspection data and then the eDNA analysis, and also the chemical sprays that happened in the farm. And then finally, we were able to develop a automated health system, the Beehive health system that is also scientifically validated, and it is kind of a dashboard so that the user, whoever the user is, a researcher or a farmer or a apiary person who can integrate, who can use this the dashboard in an interactive way to see which attributes are impacting the hive health and what happened for that kind of impact to happen in that hive. So this is just to show what kind of methods will be used to understand the data. So this is just showing a temperature kind of line graph in that data period where we collected the data from. Site 1, Site 2, Site 3, just see the Site 1 where all the hives are, the temperature is very stable. And then in the Site 3, you can see it's varying a lot and also it's fluctuating a lot. And another way of exploring the data was, that is the sound data in this one. See the first one and the third one showing a uniform kind of distribution. Not uniform, like it's uniform, but a normal distribution, but it is very spread out compared to the middle one. So it means that there are different kind of jobs the bees are doing. So in the middle one, it's very tight. So most of the bees are doing a similar job compared to the other two. So this is also using the acoustic data where we just explored the root mean square energy. So it's showing the activity, the energy activity, activity of the bees by energy levels. And then finally, this is the comprehensive kind of a dashboard Beehive health system that we were able to develop here, and the red shaded areas showing that the identified anomaly time period, and the lines, the dotted lines are showing where the chemicals or some sort of incident happened within that area of the farm. then we, once the anomaly was detected, then we did cross validation with the, to see whether there is an alignment with the human inspection data, so that you can see there is sometimes we were able to see there is a requeening activity happening, and sometimes there's a beetle found, and in other cases there were some diseases found in that hive where the detection, anomaly detection was found. Similarly, there were correlations with the chemical applications we found, and then also we were able to validate with the eDNA analysis, which were made into a sunburst diagram so that it's easier to interact with. This is showing a video how the interaction of the dashboard system works. Thank you.
Thank you very much. So for our next speaker, it's going to be Dan Ping Dong from Singapore Management University. And the presentation is titled Mining Meaning, How SMU Libraries Use NLP and AI Tools to Uncover Strategic Insights.
Okay, so good afternoon, everyone. My name is Dan Ping. I'm from Singapore National University Libraries, and today I'll be sharing 3 case studies of how our library has been experimenting around with AI tools, in particular BERT topic and large language models as well, to uncover insights from text-based data. So just a very quick background of where I come from. So SMU is a relatively young research-focused university based in the city center of Singapore. And within the libraries, I'm from the Research and Data Services team. We support researchers throughout the research lifecycle. And recently, we have been trying to develop new services supporting analysis stage of the research lifecycle, such as quantitative and qualitative analysis. So this is where we come from. Started exploring tools like BERT topic to see how they can better support our researchers, as well as the institution as a whole, because we also work on research impact analysis, bibliometrics as well. By the way, SMU Libraries will be hosting the FORCE 2026 conference next year in June. So it's another great space for discussions on open data, open scholarship, and related themes. So it will be held at SMU. So if you want to come and take a look, do submit a proposal or just join the conference. So we realized that universities, generally speaking, we hold large amounts of text data that may have interesting insights that we can learn from. So in SMU itself, we don't have a dedicated research analytics office. So the library has actually taken up this role. We work quite closely with the Office of Research, Provost Office, to come up with research metrics and impact reporting. And so we see opportunities at 2 levels. The first level is at the operational library level, is at the operational level in the library itself. So we want to better understand our library users. We also want to improve our services and also make informed decisions on new initiatives, such as something very popular recently is the AI chatbots for the library. And on another layer, we are also looking at the institution to provide bibliometrics reports to also meet our institutional needs for data-informed decision-making. So that's where we actually started to explore AI-based text analysis, such as BERT topic and also large language models. So before I delve into the case studies, I'll just give you a very quick method snapshot of BERT topic itself. So it's a Python-based AI tool for automated topic modeling. It's making use of transformer-based embeddings, which is roughly in the same family as ChatGPT and other Gen AI kind of tools. So these embeddings are very good in terms of understanding text. So BERT topic is able to group similar documents based on the meaning, not just word frequencies, which is the older methods like LDA. They're mostly based on frequency, but this one is actually based on the meaning of the text itself. It can extract the themes from large amount of text, which can be very broad in terms of its application. And another advantage of BERT topic is that it's able to work well with messy real-world data sets like library transcripts. So we talk to the users, they want to ask for a book, ask for a database. This kind of very messy kind of text is also able to interpret quite well and come up with clusters. So another advantage is that it can be run locally. So we are working with transcripts, which is quite sensitive. So we don't want to send all this data to the cloud or to OpenAI or anywhere else. So we want to just analyze this kind of thing on the local environment to ensure privacy and data control. It's also quite easy to use out of the box. There's no advanced coding required. Maybe introductory, beginner to intermediate is fine. Myself, I'm not like an advanced coder myself, but I can use it. It's relatively straightforward to use. So on the right side is the method itself. If you're interested, you can actually visit the author's website to find out more. But basically, BERT topic is very flexible. So all of these steps shown here for creating embeddings, creating clusters, and so on. So they can be replaced with other algorithms or other methods, depends on what kind of data you're working with. So our first case study was working with our library chat transcripts. As you can see from the diagram there, our library chat grows a lot during the COVID years because the libraries are closed at the time and the users have to reach out to us through the library chat. And then after that, after we begin going back to normal, we start to see a decline in the number of library chats, but still in the range of 20,000 of chat logs. So in 2024, last year, we did 2 projects. One is the operational review of chat duty deployment to optimize the resources, and also we want to understand what kind of questions do our users ask us on a daily basis. And then another project that we are looking into is to have an AI chatbot. One of our faculty member from the computer science school, she wants to explore having an AI chatbot for the library built on open source models like Llama. So we need to come up with the requirements. So we need to tell her what kind of questions usually come to the library chat. So here is the problem: is like how can we identify the key inquiry types from thousands of chat logs to understand what our user need and to understand how we can build on these new services and tools. So this is our kind of like the outputs from BERT topic. So we started with over 9,000 transcripts from 2021 to 2023. So these are clustered into about 40 topics using BERT topic. There are quite a number of parameters to tweak here and there to make sure that the end results make sense. Okay, so the table that you see over here, you see a lot of keywords, and each row will be one of the topics. You may notice the first one is a -1 cluster. That's basically the outlier group that doesn't fit anywhere else. So this is one thing about BERT topic. It doesn't force clusters to form. It only create clusters that make sense. So because we are using chat transcripts, which is quite messy real-world data, so there are more outliers in that sense. If we are using more structured data like abstract, it will be a lot lower, maybe around 10%. That's all acceptable. And within the algorithm itself, if you want to reduce the outliers, there are ways to do so. But we decide just to keep the clusters clean, and we'll just look at the nicer and cleaner clusters itself. So when we look at the results, it actually really makes sense from a library point of view. Like topic number 0. Oh my God, okay. So I'll just do it a bit more quickly. So besides getting all the keywords, we want to make the results more interpretable. So we use ChatGPT to actually try to interpret the keywords and come up with more human-readable topic labels and summaries. Like we have a group on citation, for example. We have another group on academic database access, and so on and so forth. So out of all these groups, we also try to group them into higher categories. So these are the groups of queries that we have found. So what's the practical implications here? We basically use the results to inform the 2 projects that I mentioned earlier on to optimize our chat duty deployment and also to contribute to an AI-powered chatbot. And there are also lots of visualizations within BERT topic itself. This is one of it, to visualize the different clusters, which is it does make sense to us working in the library. So case 2, we are using the same method, also BERT topic, but this time we want to discover the themes of research done by SMU researchers on Asia. So this is in collaboration with our Office of Partnerships and Engagement. We basically try to get all the faculty publications on Asian countries, about 900 in total. Then we run BERT topic on the title and abstract to extract the themes of these papers with Cloud and ChatGPT to generate the human-readable labels. So again, these are some examples. We discovered 10 Singapore-focused themes and 12 non-Singapore Asia-related themes, and mapped them into the SMU's research focus areas. So some lessons and reflections from this exercise, we realized that when we are communicating with higher management, they prefer clear story-driven summaries more than keywords. Keywords doesn't really make that much of sense to them, so we try to use GenAI to create these more readable summaries. And these are topics of strategic importance as well. We're trying to provide research intelligence and evidence for strategic decision-making. So the next one, very quickly, is how we use GenAI for thematic classification. Again, this is another question from our research office. How can we classify the publications under our key pillars, the digital transformation, sustainable living, and growth in Asia? So we basically run the titles through the DeepSeek API or Cloud API, depends on which tool we have access at the point of time. So we basically did the zero-shot classifications. They assign the categories based on just the definitions, and no training examples is provided for this kind of publications. And it works pretty well, as we found out. So basically we find this quite reliable, but we also noticed that sometimes it can be subjective. So management may think in a certain way, but the large language models may not be aligned with what they think. So we do have to do it back and forth, tweak the prompt a little bit, because the even small wording changes can actually impact how the publications are categorized. But we also realize that even you ask different humans to categorize, sometimes they may have different conclusions. So it's just a matter of tuning the prompt until it actually meets our requirement. Yeah, so there's a certain degree of subjectivity there. Last slide, yeah. So basically, we realized that AI tools are now a lot more accessible, and we want to rely on these opportunities to showcase our value in the institution for strategic decision-making. Yeah, so thank you.
Thank you. So the next speaker is Robert Grossman from the University of Chicago with the paper Accelerating Research with Data Commons and Data Meshes. And of course, his entire team he will present. Thank you.
Well, to keep you entertained, instead of one 10-minute talk, I'm breaking this down into three 3-minute talks. I'm going to do the first 3 minutes. So I'm going to tell you about the Gen 3 data platform. It's a data platform that's open source and can be used to create data commons and data meshes. And in the roadmap over the next 6 months, after only 2 years of work, we're putting AI services into it. The core idea of a Gen 3 Commons, and there are about 30 or 40 of these around the world by about a dozen organizations. The largest one has about 100,000 users a month with 3 to 5 petabytes of data pulled out each month, but it scales down to a few dozen to a few hundred users per month. The idea of a Gen 3 data commons is very simple. You create a data model, and then the code itself auto-generates fair APIs for structured data, for data objects, for semi-structured data, and in the next release for vector objects to support AI-based embeddings. And then it also auto-generates a data portal and auto-generates a data portal for submitting data. And there are attached workspaces so you could analyze the data. The data, Gen 3 also creates data meshes. We have about 4 of these in production. And this is the ability to bring together multiple from, we have up to about 15 to, I guess, about 20 data platforms, both Gen 3 and non-Gen 3 platforms that interoperate to create a data mesh. And then there's a central hub where you could search and discover data, and then there's interoperability that's provided. And the core idea is to do this over a set of a handful of services called data mesh services. This is just a picture we have 4 data meshes at the top. We have about a dozen data commons that we and our partners operate. Then there are about a dozen of these around the world, operated by third-party organizations, primarily in the US and Australia and New Zealand. So apropos of that, I'm going to pass it to Claire Rive from the University of Hull.
Thank you. So I'm going to very briefly, I guess in 3 minutes, touch on one of the use cases, or a couple of use cases for Gen 3. I'm going to start at the back and thank everyone. So this is both of the repositories I'll briefly mention are very collaborative work. So this is the team really behind the Aotearoa Genomic Data Repository and are actually in a parallel session presenting on it at the moment. And likewise, Rakaiora is a huge collaboration, and Phil Wilcox is around somewhere and is also presenting. But really what I wanted to just briefly mention was that we have run 2 Gen 3 platforms, and we, I guess in an Aotearoa New Zealand sense, have quite a different perspective of what open data means and what data sharing means, and have been actually able to utilise the Gen 3 technology in order to have a much more controlled access data commons in a couple of different examples, and I'm very happy to talk a little bit more about that. Ordinarily, I would probably start a talk with the context, so apologies, I'm literally doing this a bit backwards, but in a New Zealand setting in Aotearoa, the Te Tiriti o Waitangi, or the Treaty of Waitangi, kind of sets the grounding for how we consider working with Indigenous data, and that includes genomic data, so in the context of these 2 projects, both human and non-human species. There's already been a lot of talk on sort of the care principles in Indigenous data this morning already, and I guess just to highlight in the last 10 seconds a number of frameworks from a Aotearoa New Zealand perspective that ground that work in data sovereignty. And I'll now pass over to Bernie. Thank you.
Thanks, Bob and Claire. So I'm going to talk about some work we've been doing in Australian Biocommons. A few years ago, many organisations in Australia got together along with Australian Genomics, and they looked at the data sharing landscape for humanomics data in Australia, and they found that Australia is imminently facing a crisis in genomic information storage, analysis and sharing. And so one of the major things we're doing in Australian Biocommons is addressing this particular challenge. And this is the vision that we're aiming for from a user perspective, a research user. It covers the whole gamut of FAIR, and in particular, people can find the data, they can explore the data sets, they can, if they're interested in the data and it's appropriate for their research, they can apply through appropriate governance data access committees. If it's approved, then they can take it to appropriate scalable computing resources to analyse the data. And we're doing this with Gen 3 in a number of situations. This one here is the leading program of work that we have at the moment. It's MRFF funded. It's called the Australian Cardiovascular Disease Data Commons, or ACDC, and it's Australia's first human multi-omics data commons. We're aiming for 18 harmonised cohorts relating to cardiovascular disease with about 400,000 study participants, or about 100,000 of those with genomic and lipidomic data. The reason this is important is because you probably know that cardiovascular disease is a major cause of mortality and morbidity in Australia. About 27% of individuals who present with a heart attack at hospital have no prior clinical indicator of that event. That means that the first time they're seen by a doctor is when they have a heart attack, and clearly that's no good, because there's no chance for intervention to avoid that event. And our colleagues in the Australian Cardiovascular Alliance and CAD Frontiers have brought the community together to assemble the data to actually solve this problem and identify new markers for preclinical cardiovascular disease. So that's why we're building this data commons. Just conclude here with a few final remarks that Stephen Manos, who leads the BioCloud team at Australian Biocommons, and that team is the technical team that delivers the Gen 3 platform for us. I don't have time to go through all of this in great detail, so I'll just point out some highlights. Stephen mentioned that we have a small human data focused Gen 3 team that sits within the Biocommons project. We have about 3 FTE to develop this data commons, and including 1 FTE of cybersecurity. So we have a small core team, but the great thing about Gen 3 is when you have established this platform, you can redeploy for new projects fairly quickly and efficiently. And that's what we're aiming for. We're about to have about half a dozen of these data commons in different research domains coming on board, pending funding outcomes, but hopefully that will be in the next couple of years. And here are some of the lessons learned. One thing is we have worked very closely with our research communities, and the data user stories drive the implementation. So we think very carefully about the research questions that people are asking of their data when we're designing the data commons. We need to establish foundation infrastructure, both common and deployment specific. The data commons also link to many other infrastructure that we're developing, including identity and access management across Australia for humanomics data sharing and analytics platforms, and we tie all of these foundational infrastructure together. Security is obviously paramount with human data. I don't need to say that. You guys all know that, and obviously we think about that very carefully. And lastly, building communities around these data commons is really critical. Ultimately, the communities are the users, and they drive the requirements for the development of these platforms, and ultimately determine the success by taking the data in these commons and turning it into translational outcomes from research. And that's the last slide. Thank you.
Thank you very much. And the last presentation of the session will be done by Daphne Raban from the University of Haifa, Co-Data Israel. And the title is Data-Driven Risk Identification in Supervision Reports of the Ministry of Health. Oh, sorry. Yeah. How do I switch with the arrow? Yeah. Thank you.
Thank you.
Yeah, sorry.
So good afternoon, everybody, and thank you for staying this late. So I'm going to present this work that I've done together with Avital Tzadok from our Ministry of Welfare. Towards the end of the 20th century, industrialized nations confronted what was called a regulatory crisis. Regulation was criticized as excessive, rigid, and costly. And in response, governments began searching for ways to make it more effective. In the United Kingdom, for example, under Margaret Thatcher, they introduced a new public management approach, borrowing from the private sector practices and rendering or redefining government's role from a service provider to a supervisor. And by the late 1990s, again, according to private sector practices, risk identification and risk management emerged as a key tool in supervision to be done by the government. So again, the government has transformed from actually doing the job to regulating and overseeing and checking what is done by outsourced service providers. Many countries followed suit after the UK, and Israel as well has adopted these ideas, and they gain prominence as part of efforts to reduce the regulatory burden in the country. So since 2014, a government program emphasized balancing the risks of under-regulation with the costs of excessive intervention. So here too, risk management plays a central role, and what is being done is actually the government sends supervisors to examine what is being done in the field. But the problem is that these supervisors write very long and detailed reports that don't say that I said this, nobody reads them. So then if nobody reads them, then what can we do about them? So here we come in, Vital and myself, and we thought we could implement AI in order to conduct risk analysis and be able to fish out the risks out of the reports and highlight them in order to make the supervision more cost-effective and more effective in general, not just cost-effective. So this study is actually more of a methodological study where we wanted to see if we are able to identify risks, to classify the risks, to evaluate the models. We compared 2 different models and to find the practical application. And so the data we selected, or the data we were able to put our hands on, because it's quite difficult to gain access to data. I mean, the whole conference is about this, but it's quite difficult to gain access. So we found supervision reports of the health ministry in Israel, and we downloaded these publicly available reports, and we randomly selected 400 sentences out of these reports, so that 200 sentences were labeled by us as being delivering risks or describing risks, and 200 sentences that were not describing risks. Okay, so we had 200 negative and 200 positive kinds of sentences, and we prepared the text for NLP analysis, and then we applied 2 different models. One is the Hebrew version of the well-known BERT model, and another is also another Hebrew version of the BERT model, which is called Dicta. It's just trained on a different set of data. And so then we took those 400 sentences, we ran a learning or training session, then we ran a validation session, and then we ran the test session, and we checked for 2 different, we did 2 different kinds of tests. I'll describe that in a second. So first, just to give you an example of what is classified in those 200 risk-describing sentences. For example, a sentence such as, there was no publication of the payment ceiling for dental treatments. Okay, so this is a negative inclined sentence versus on the right side we have the more neutral or positive sentences, such as doctors are familiar with the entitlements in the dental field. So this is a more positive sounding sentence. So we first tagged the sentence manually in order for the machine to be able to later do the test. So after we ran these 2 models, what we did first was to test whether they found correctly the risk-describing sentences versus the neutral/positive sentences. And then we categorized all the sentences or all the risk-carrying sentences into 5 categories. One was the administrative procedures, and there is an example here. I don't want to read it to you because of time constraints, but you can see the example. So that was the first category. Then we had a category devoted to the medical staff, the medical services, infrastructure and equipment, and finally patient rights. Those are the 5 categories we were able to identify as types of risks in these health supervision reports. So when we tested to see the efficiency or the effectiveness of the 2 different models, the Hebrew BERT and the Dicta model, we saw clear advantage for the Dicta, which is here in blue. It scored better on all 3 quality scores for the comparison between the two models. So dicta was preferable for finding out whether a sentence was negative or not. And then it also performed better than the BERT, the Hebrew BERT model, in identifying the 5 classifications. So overall, this dicta model emerged as our favorable or our better model for the entire job, for distinguishing positive and negative and distinguishing between the 5 categories. And so, first of all, we've shown that AI can effectively identify risks in unstructured regulatory texts. So we can deliver it to non-readers in a very effective way. Dicta is the superior model in both tests. We do need the human judgment first for the training and then also for establishing the categories. And of course, the practical applications are enormous. It could be health, it could be other government services such as welfare services and a whole host of other kinds of education, a whole host of government services that, that deliver lengthy reports that nobody reads. Okay, so the limitations are, of course, our set was quite limited, quite small, but still, on the bright side, we had a small set, but we were able to achieve very strong effects. So it would be better to increase the scale of the study in a future work. And also another limitation is that we limited ourselves this time only to healthcare, but we could, as I said, go to other fields of government activity and to test our models there. So in the future we could do that, and we also need to keep a mindful eye on ethical concerns, of course, when we employ machines in order to help us in reading supervisory reports. So I hope I managed to keep the time frame. And here are emails, and we're happy to be in touch, happy to discuss. And thank you very much.
Thank you very much to all the presenters of these really excellent papers. I know we are out of time, but maybe we can take a few questions still and go a bit into the coffee break. So if you have a question, please do raise your hand and I'll come with you with the mic. Anyone? I'm sure you have a question. We have 8 presentations, so if not, then yeah, no. Everyone who wants to go to the coffee, yeah, right? Anyway, yeah, go and fetch the presenters during the coffee break and ask them more questions, of course, yeah. Yeah, we were trying to keep time, but it always takes a lot. It's too much to have 8 presentations in one session, I believe, but okay. Thank you again for joining.